So, Mark, currently I am doing my research internship at IIT Guwahati. The topic of my research internship project is molecular topic, which I want to present here. For that, for presenting this, I want to tell you about background of it, why I am doing that. As we know, the E. coli is a bacteria. It is present in our guts as well as some pathogenic E. coli bacteria, which cause disease that harms us. So, it present in water or in food sample. Therefore, it is essential to encounter or identify that is the water is contaminated or the food is contaminated we are consuming. For that, we have to identify that how we have to identify is there any bacteria or E. coli present in our food sample or water sample. So, for this, uh, some research works are going on. And as we know that every living organism made of protein, so E. coli bacteria, similarly, E. coli bacteria have a specific protein known as beta glycosidase, okay, which is present specifically on E. coli bacteria. That can we can which we can use it for the reaction with CPRG, red chlorophenol beta glycopyrolusite. This is a ligand and protein is beta glycosidase. Okay? Then after that, what happens when they react? Some reaction have going on, bone form, some aromatic ring form, hydrogen pi interaction form. So that part I have to visualize and present in front of you. That part is comes under the domain of molecular docking. And let me tell you about what is molecular docking. Molecular docking is nothing but identifying the most stable active sites where ligand can interact with protein and binds with it. So that part is comes under falls under the domain of docking. So in the presentation, I will show you more about visualization part as comparison to textual context. So let's move on. And first, I have mainly in this PPT, I have documented every step which I follow to get my desired result, animations or image for the reaction or interaction going between the protein and the ligand. Okay, let's go forward with it. This is the structure of E. coli bacteria. Okay. Uh, it is a pathogenic bacteria. Some E. coli bacteria present in our guts, which are helpful for our body, and some its types and species are pathogenic in nature, which cause various diseases. Okay, and these are the some diseases which caused by this bacteria, like gastroenteritis, which is similar like a diarrhea, cause diarrhea and vomiting. It gets through from contaminated food and water. Another disease called urinary tract infection (UTIs), and most common cause of bladder and kidney infection. Third one is uh, neonatal magnetis, brain and spinal cord infection in newborn babies. And another one is sepsis, which is caused in a blood infection, can lead to septic and shock like treatment. So this is the data which show us the death was death or mortality rate of the people, the person who are suffering from the disease called mortalities, uh, stream infection, uh, due to which a person die after 30 days. This bloodstream infection, which is also caused by the similar bacteria, E. coli bacteria. The female rate is as much more higher as comparison to male one, and you can see with age category. And another thing is known as a uh, magnetis and neonatal sepsis, which is a second largest infection killer under 5 years. So it is essential to encounter this. For that, research are going on. And now my domain comes here and my project comes here. So I begin from here. My project is molecular talking, which I will show you the steps I followed and get the desired image, result, and animation or 3D picture of interaction. So first steps is to prepare preparation of the protein structure. For preparation of the protein structure, I have to download the protein 3D structure from PDB, protein database kind, from the Google, which are available as in open source. I downloaded, which look like this one. This is the whole. This is the structure of protein, and shows it protein database bank and format of the image or the 3D structure I downloaded is PDB format, and software used for the visualization or the image visualization or image creation is Python. Is protein ID is 3 which you can type in Bible and can download directly from there also. And in 3D it looks like this one, which I have made a small clip for you. This is the 3D structure, how the protein look like in 3D. And after that, this is the steps I follow on for the preparation of protein. I go in the Bible and open the protein I have downloaded and follow the following command. As we protein have mainly four monomer chain. So for identification of four or distinguish between the four monomer chain, I input this prompt command in the Bible to distinguish between them. For chain one, I color red. For chain two, I color blue, and for chain three, I color green. Likewise, for chain D, I colored yellow. It looks like this. After that, this one, the red one is chain one, blue one is chain two, and C is green one, chain three, and D is chain four. After that, some atoms, other things are also present in protein like water molecules, dimethyl sulfoxide, magnesium, sodium. So it is essential to remove those molecules to prevent from hindrance create during the interaction of the protein and the ligand. So I put the following command like remove RESN stand for residual, remove residue dimethyl sulfoxide, remove residue sodium, remove residue magnesium, remove residue hydrogen, and also eliminated some of the other 149 some compounds present in the protein. Removed some other chain like B, C, and the chain A only. Get to know how the reaction are going in a unit cell. So I performed all everything on monomer structure. Same thing happened at every monomer of the protein. Okay. This is the protein after the removing of dimethyl sulfoxide, sodium, magnesium. This is the 3D look of the protein, which you can. After preparation of the protein, we have to set the protein molecules in PDB format. So for that, I use the software known as Autodogbina and remove the water molecules, add polar hydrogen, and add gastrin charges to support interaction and for better interaction between ligand and protein. And saved as a PDB format. This is the image which I take a screenshot from there. And this is the 3D view. You can look upon it how it looks like after the preparation of the protein. It move on. And similarly, same thing I have to do with the ligand part. I have downloaded the ligand from the open source available in the internet. Uh, and the ligand is a chlorophenol red beta glycopyranoside. This is its 2D structure and this is 3D. And you can, for visualization, I have put a small clip for the ligand one. Uh, green part is chlorine molecules. Uh, this parrot green is carbon one and white one are hydrogen, red are oxygen, and that yellowish one is sulfur, which is present in the ligand known as beta glycopyranoside. 3D animation. And as I have told that we also have to prepare the ligand in a similar way as we have prepared the protein. So I will follow the same the path or the steps for ligand one, ligand two. Remove the water molecules, added for the hydrogen, added gastrin charge, detect torsions, save it as a PDBQT format. And after that, we have to create a configuration file where of the molecules where we are for the interaction, and this is the configuration of the protein and the ligand. Basically I am following a blind docking, so the center which I have put here are copied from the docking grids, which I will show in further slide. The size of the docking box is 40, 40 X, Y, and Z axis. Energy range I kept here is for you can keep according to your need. This is the docking which I have copied the same data and put in a txt file. And same thing I have copied and put there on txt file. This is the grid box. Grid box basically have here we perform to know where the ligand will go to interact with the protein for identifying the active sites of the protein molecules. And after that, I have
completed everything, protein PDB format, ligand PDB format, application Autodoc Vina, and the TXT file which I have showed you of the grid data in a file, and after that I have to input the command prompt for the docking processes. Uh, s- uh, these things I have followed cd data, cd dot, and after that I have copied the path of the file where I kept everything in the file. This is that one. And after the then my next step is to verify and check is all things are there in the file or not. So for that I input the command directories dir, and we can see the following files contained in following files are there in that folder, now which I kept name as a summer sub project. And this is the configuration text file. This is the main ligand PDB format, and this is a protein monomer PDB format, Autodoc Mina, which is software. And after that, after inputting the command prompt, I get the docking result. And here we can see the affinity kilocalorie mole is minimum in first one, which is the most stable side, and these are others which are due to the difference in their orientations or configuration, we can say, due to the orientation. And this is the most stable one, so I will go with that part only. And this is how after docking the interaction will can we can visualize in 3D look like this. This is the active site interact with the protein. Here in this 3D clip, I haven't showed the how interaction are going on. Just I have showed you the where ligand interact with the protein. In further slide, I can show you how the bonds, which bonds are formed between protein and ligand. So let's move forward. And this is the 2D image, how interaction are going on. And here I want to tell something to you. This is the ligand, and this is the part of the protein which are interacting with the ligand. And these green dash lines represent the hydrogen. Red R, this red R, are the hydrophobic contact with the protein molecules. And blue circle, this one, this is a polar atom, polar atoms. And black rings are aromatic interaction. This one, you can see 2D image. For its 3D look like this, you can see this one is ligand, and the rest part are the protein part which are interacting with the ligand. And this is the name the part of arginine, tyrosine, aspinin, which are present in the protein. For this, I use software called Python, and a short clip where the protein molecules are present without the removing of the interacting part. This is another short clip. You can see the protein. without this receptor protein. Here, the rest part of the protein part have been removed for better visualization and better presentation. This is the ligand molecules. This is the part of the protein which are interacting with the ligand one. You can look upon. I will explain which what is the name of the bond here interacting with the ligand part. What is the name of this in further slide. So basically, you can see how it looks like. Forward. This is the data. As the green lines are ligand non-bond monitor, and light greens are ligand non-hydrogen bond, uh, carbon hydrogen bond, and the green one is covalent hydrogen bond, and the purple pink one is pi alkyl bond. This is a better view of that one. I use software study discovery visualizer for creating this simulation. This is another view where I make the ligand part in ball and stick format and the other part in this one is ligand, this is pi bond, this is the bond length, and this one is carbon conventional carbon hydrogen bond. This is a carbon hydrogen bond green line. The black lines are conventional carbon hydrogen. I have mentioned here the color coding. The black one is carbon conventional carbon hydrogen bond. The green lines represent the carbon hydrogen bond. And the pink one is hydrophobic pi alpha bond. This is